Welcome to Lecture 12.1, in which we will introduce how expression of genes is regulated, beginning with prokaryotic gene regulation. Our learning objectives for this video are to understand how prokaryotes regulate gene expression, define activators, repressors, inducers, and operons, and describe how the lac operon works. Let's take another look at the central dogma of molecular biology. Our genetic information is stored as DNA, and when a cell wants to make a protein using the instructions, it will transcribe a complementary copy of mRNA from the DNA and then translate the mRNA into an amino acid sequence. A gene is expressed when the DNA is opened up to make RNA and proteins. The thing is, there are a lot of genes, and we don't want to express all of them all the time, as that would require far too much energy, and most genes are simply not needed all the time. That's why regulating gene expression is very important. While gene expression is usually regulated at the transcriptional level in prokaryotes, it can be regulated at the transcriptional, post-transcriptional, and post-translational level in eukaryotes. We will first go over transcriptional regulation in prokaryotes. Prokaryotes are unicellular and have relatively simple genomes. For them, the purpose of gene regulation is to help the prokaryote respond to environmental cues, such as availability of nutrition or stress. Control at the transcriptional level can be either positive or negative. Positive regulation is when an activator binds to the DNA, usually at the promoter of a gene, to increase transcription. This arrow represents the transcription start site, and the RNA polymerase is recruited by the activator to perform transcription. Negative regulation is when a repressor binds to the DNA, usually at the operator of a gene, to decrease transcription. Operators are regulatory DNA sequences found between the promoter and the gene, and are mostly only found in prokaryotes. When a repressor binds to the operator, it prevents transcription by physically blocking RNA polymerase from transcribing the DNA. An X is used to mark that there is essentially no transcription. An inducer is a molecule that signals changes in the environment to the cell, and can either bind to an activator to increase transcription or bind to a repressor to decrease transcription, depending on what the cell needs. An operon is a block of several genes next to each other that code for proteins that work together to perform some function. The most famous example of gene regulation in prokaryotes is the lac operon, which contains genes that make proteins involved in using lactose as an alternative energy source when the main energy source, glucose, is not available. This includes LAC-Z, which codes for beta-galactosidase, the enzyme that breaks lactose down into glucose and galactose, LAC-Y, lactose permease, a transport protein that allows the prokaryote to take in lactose, LAC-A, lactose transacetylase, which adds an acetyl group to lactose, and LAC-I, which codes for the lac repressor and has its own promoter, separate from the other three genes. By default, the lac operon is off, although there is usually a very low amount of transcription. The lac operon will only be fully active when there is little to no glucose available in the environment, and when there is lactose available. After all, there is no point in dedicating energy to making proteins for a non-preferred nutrient if the preferred nutrient glucose is available, and also no point if there is no lactose to process. The lac operon is kept off by the lac repressor, made by the LAC-I gene, binding to the operator. If lactose is available, a metabolite of lactose, allolactose, is produced by the small amounts of proteins being produced by the lac operon. Allolactose then acts as an inducer by binding to the repressor, changing its shape so that it can no longer bind to the operator, and inducing activity of the lac operon. Once lactose levels fall, allolactose levels would also fall and stop binding to the repressor, and the repressor would bind to the lac operator again. While removing the repressor increases transcriptional activity, lactose and allolactose will not be able to fully induce lac operon activity if glucose is present. In addition to removal of the lac repressor, the lac operon also needs the presence of an activator. This activator is called CAP, which stands for catabolite activator protein. This is a protein that binds to promoters of operons that are involved in processing alternative nutrients. CAP itself is regulated by cyclic AMP, a nucleotide involved in signaling. Alone, CAP does not bind DNA well. With cyclic AMP, its shape changes so that it can bind to DNA and recruit RNA polymerase for transcription. High glucose levels in prokaryotes decreases cyclic AMP levels, which prevents CAP from being able to bind to the lac promoter and activate transcription. However, when glucose levels fall, cyclic AMP levels increase, and this activates CAP, which in turn activates the lac operon by allowing RNA polymerase to better bind to the promoter. Of course, if lactose levels are low, and the lac repressor is still on the operator and blocking RNA polymerase, the lac operon would still not be active. This is how prokaryotes ensure that the lac operon is only fully active when glucose is low and lactose is high. Glucose itself is also able to repress the lac operon by inhibiting lactose permease, the lactose transporter made by LAC-Y. This makes it more difficult for lactose to be transported into the cell, even if it's present. This table summarizes the activity of the lac operon under different conditions. When there is plenty of glucose but no lactose, the lac repressor is bound to the operator and there is no transcriptional activity. When there is no glucose and still no lactose, cyclic AMP levels increase and CAP binds to the promoter, but the repressor remains bound, so there is no transcriptional activity. When there is glucose and lactose, the repressor is no longer on the operator, but there is no activation from CAP, so there is low transcriptional activity. When there is no glucose and high lactose, the repressor is gone and CAP fully activates transcription. In summary, we learned that prokaryotes regulate their gene expression by controlling transcriptional activity in response to changes in the environment. Activators are proteins that bind to the promoter to increase transcriptional activity. Repressors are proteins that bind to the operator to decrease transcriptional activity. Inducers are molecules that signal changes in the environment to the cell so that it can increase or decrease transcriptional activity of genes. Operons are groups of genes that code for proteins that work together. In the lac operon, CAP with cyclic AMP acts as an activator by binding to the promoter, the lac repressor binds to the operator to block RNA polymerase, and allolactose, made from lactose, acts as an inducer. In the next video, we will discuss eukaryotic gene regulation. Thanks for watching.